Yes, start, madam.
Okay, sir. Good morning, students. Welcome to another new session of molecular biology. Welcome to Telangana Residential Educational Institution Society. Myself, Sarala, working as a botany lecturer at Hasanparthy, Warangal. Yesterday, we already learned about the structure of DNA and the replication of DNA. But few points in the replication are not completed in the yesterday. So that's why I once again recapitulate the DNA replication. Okay, DNA replication. What is DNA replication? The DNA molecule, it having the ability to produce two daughter DNA molecules. This process is called as DNA replication. DNA replication means one DNA molecule which is having the ability to produce two daughter DNA molecules. They are always identical to their parental DNA molecule. And this DNA replication process is catalyzed by an enzyme DNA polymerase. So once you already observe this diagram in the previous class, once again the students observe this diagram. So this diagram clearly explains the DNA replication. These two strands are there in the DNA molecule, and one strand is runs in 5 prime to 3 prime direction, and another strand is runs in 3 prime to 5 prime direction. And in this replication process, DNA polymerase enzyme, it can place an important role. It is the most important enzyme which is involved in DNA replication process. Okay, and this DNA replication process is takes place in the S phase of interphase in the cell cycle. And this DNA replication as well as cell division both are coordinate with each other. And this DNA polymerase enzyme catalyzes the total polymerization in the replication process. So along with this DNA polymerase enzyme, some other enzymes like DNA ligase, this enzyme also plays a vital role in the replication process. And this helicase enzyme also plays an important role in the DNA replication process. This helicase enzyme, it can unwind the double helix of DNA. DNA molecule present in double helical structure. So these two helix are separated, or these two strands are separated with the help of this helicase enzyme. Helicase enzyme causes the unwinding of DNA molecule, and this unwinding, it can forms a replication fork. By the formation of the replication fork, by the formation of this replication fork, the bases which are present on the two strands are exposed and they are involved in the synthesis of new strands. This helicase, it is very, very useful for this unwinding of this DNA double strand. And in this DNA replication process, RNA primer nothing but a short piece or segment of RNA molecule, which is very, very necessary for the synthesis of two daughter DNA molecule. Without DNA or RNA primer nothing but a short segment or piece of RNA molecule, which is initiates the DNA replication process. Okay, and DNA replication is initiates at a specific site. That site is called as ori site. DNA replication which begins at a specific site. Specific site means ori site. Ori site means origin of replication. DNA replication is begins at this specific site. This site is called as oocyte. This oocyte is differ from one organism to another organism. And so once replication process is start, and this DNA polymerase enzyme is bind to this DNA polymerase enzyme is bind to the RNA primer and it initiates the replication process. Okay, and this DNA molecule, it consists of two strands, but in this two strand only one strand is involved in the replication process. Later, after some time, another strand is also involved in the replication process. But two strands are not simultaneously involved in the replication process. First, one strand, that is this strand 5 prime to 3 prime runs in this strand. This strand is involved in the replication process initially, and this strand is called as leading strand. Leading strand means the DNA strand which is first involved in the replication process. This strand is called as leading strand. After some time, another strand is involved in the replication process, and this strand is runs in 3 prime to 5 prime direction. This strand is involved in the replication process. After some time, this is called as lagging strand. In this lagging strand, the newly synthesized DNA is formed in small fragments, and these fragments are called as Okazaki fragments. Clearly listen here. In the case of lagging strand, the newly synthesized DNA is developed in a small fragment. This is one fragment and this is one fragment. So these small fragments are called as Okazaki fragments. And in this lagging strand, replication is takes place discontinuously. So that's why the newly synthesized DNA is formed in a small fragments. These fragments are called as Okazaki fragments. After the completion of the replication process, these fragments are attached with each other with the help of DNA ligase enzyme. So this is DNA ligase enzyme. It can attached or joined the small fragments of DNA, and due to the ligation, these fragments are attached with each other. They can form a complete strand. Okay, and in this manner, DNA polymerase is moves randomly, and this helicase enzyme is also moves randomly, and it can cause the unwinding of DNA molecule. This RNA primer and helicase enzyme, DNA polymerase enzyme, these three, they can move randomly step by step, and they can cause the unwinding or uncoiling of DNA molecule, and that replication process is takes place continuously. Okay, and replication process is begins at a particular site. That site is called as ori site. Ori site nothing but origin of replication. Thus each DNA strand as act as template for the synthesis of new strand of DNA. This reaction is catalyzed by DNA polymerase enzyme. This causes the chain to elongate, but it cannot start the formation of a new strand. DNA or RNA primer is essential for the initiation of DNA replication. This RNA primer, it is very, very essential for the initiation of replication process. Primer nothing but a short piece of DNA or RNA is considered as primer. A short piece of DNA or RNA is used as primer for the initiation of DNA replication. Replication occurs within a small opening of DNA helix referred as replication fork. The DNA polymerase, it catalyzes the polymerization only in one direction, that is 5' to 3' direction. On the one strand, the replication is continuous. This strand is known as leading strand. This strand is known as leading strand. And in the other strand, replication takes place discontinuously. This strand is known as lagging strand. So leading strand means replication takes place continuously. This strand is known as leading strand. And in the another strand, replication takes place discontinuously, and this strand is known as lagging strand. The discontinuously synthesized fragments are known as Okazaki fragments. These fragments are later joined by the enzyme DNA ligase. So these are the two strands. One strand is runs in So one strand is runs in 5' to 3' direction, another strand is runs in 3' to 5' direction. So in this strand, it is replication fork. So by the activity of the helicase enzyme, two strands are separated from each other. So DNA molecule which is present in the double helical structure, but at the time of replication, these two strands are separated from each other. They can form this replication fork. By the formation of the replication fork, the bases which are present on the two strands are exposed and they are involved in the replication process. So these two strands are called as template strands. The replication is first begins in this strand. This strand is called as leading strand. And the DNA polymerase enzyme, it can catalyze the total replication process. And this strand is laterally involved in the replication process, and the newly synthesized DNA is developed in the form of small fragments. These small fragments are called as Okazaki fragments. After the completion of the DNA replication, these Okazaki fragments are joined with each other with the help of DNA ligase enzyme. And this strand is called as lagging strand. Lagging strand means this strand replication starts somewhat later. So that's why this strand is called as lagging strand. And in this lagging strand, the newly synthesized DNA is formed in the small fragments. They are called as Okazaki fragments. And this is total structure is replication 4. This total is replication 4. This DNA replication process is very, very essential process, and which takes place in the S phase of interphase before cell division. Once DNA synthesis or DNA replication is completed, the cell is ready for cell division. Okay. And this DNA replication takes place through semi-conservative method. Semi-conservative method means, so this is the parental DNA molecule. And this parental DNA molecule is involved in the replication process through semi-conservative method. Semi-conservative method means Due to this replication, one DNA molecule having the ability to produce two daughter DNA molecules. In this two daughter DNA molecule, each daughter DNA molecule consists one parental strand and one newly synthesized strand. Another daughter DNA molecule is also consists one parental strand and one newly synthesized strand. At every generation, each daughter DNA molecule consists one parental strand compulsory, which means the parental strand is transfers from one generation to another generation. That genetic information which is present in the parental strand is preserves for other generations, and this mode of DNA replication is called as semi conservative method. Okay, and this semi conservative method of DNA replication was experimentally proved by measles and Stahl. Okay, and this semi conservative method of DNA replication was clearly experimentally proved by Michelson and Stahl. So next, let us discuss about the experimental proof. This experiment was conducted by Michelson and Stahl. These two scientists, they conduct this experiment. So in this experiment, it is now proven that DNA replicates semi-conservatively. Semi-conservatively means one parental DNA molecule is involved in the replication process. It can produce the two identical copies of daughter DNA molecules. And each daughter DNA molecule consists one parental strand and one newly synthesized strand. This mode of DNA replication is called as semi-conservative method of DNA replication. It was experimentally proved by Michelson Michelson and Stahl. Another scientist is Stahl. These two scientists are biochemists. They conduct experiment about DNA semi-conservative replication method. So in this experiment, these two scientists select the E. coli bacteria. E. coli means Escherichia coli. So in most of the experiments, scientists use this E. coli bacteria because its cell structure is very simple, and every 20 minutes it undergo binary fission and it increases its progeny within the short period. So is it clear? E. coli bacteria, it undergo binary fission to produce two daughter E. coli cells. Means the division period is very less and the cell structure is very simple. So that's why this E. coli bacterium is used in many experiments. They grow the E. coli in the medium containing NH4Cl. NH4Cl means ammonium chloride, and as the only nitrogen source for many generations. They have to select the E. coli bacterium, and that E. coli bacterium is cultured in the ammonium chloride medium, and this medium is having N15 isotope. It is the N15 isotope, and this medium is containing ammonium chloride with N15 isotope. It is the only nitrogen source for the growth of E. coli bacterium. Next, the result was that the N15 isotope was incorporated into newly synthesized DNA molecule. So this is the experiment. So these two scientists, Michelson and Stahl, they choose the E. coli bacteria for their experiment. They have to grow the E. coli bacterium in NH4Cl medium with 15 N15 isotope. This one is normal DNA which is present inside the E. coli bacterium. They have to cultivate the E. coli bacterium in this N15 isotope containing medium. So after 20 minutes, this E. coli bacterium undergo binary fission and it can produce two daughter DNA molecules. One DNA molecule having the ability to produce two daughter DNA molecules that requires only 20 minutes time. So in these two daughter DNA molecules, each daughter DNA molecule having one N15 isotope containing DNA and another strand having normal DNA. Okay, which means the result was that the N15 was incorporated into the newly synthesized DNA. This heavy DNA molecule could be distinguished from the normal DNA by centrifugation in cesium chloride solution with density gradient. This N15 isotope is incorporated into one DNA strand, another DNA strand having normal N14 normal nitrogen molecule. This heavy DNA molecule is distinguished from the normal DNA by centrifugation process with cesium chloride against the concentration gradient. So then they transfer the cells into a medium with normal NH4Cl and took the samples at various defined time intervals as the cell multiplied and extracted the DNA that remained as double-stranded helix. So once these cells are separated from the N15 medium and they are growing in the N14 medium. So these two DNA molecules having one N15 strand and with normal N14 strand. Thus, the DNA that was extracted from the culture one generation after the transfers from N15 to N14 medium. After the 20 minutes, that had a hybrid or intermediate density. So next, the DNA extracted from the culture after another generation was composed of equal amount of this hybrid DNA and the light DNA, nothing but normal DNA. So E. coli cells are cultivated in the medium containing ammonium chloride, that is nothing but NH4Cl with heavy isotope N15. So it is the heavy isotope of nitrogen. So next, these cells are centrifuged with the help of cesium chloride and identify the heavy DNA molecule. Then this E. coli cells are again transferred into normal medium with normal N14 nitrogen molecule. And after the completion of the one generation, we have to observe the DNA molecules. These two daughter DNA molecule having one N15 isotope and another strand with N14 isotope. This red color is with heavy isotope and this green color is normal nitrogen molecule. So after the completion of the one generation, when we observe the second generation, I previously explained the E. coli requires 20 minutes for binary fission. It divides very rapidly and it increases its progeny within the short period. After 20 minutes, it can give two cells. After 40 minutes, it can give four daughter cells. So these four daughter cells having four DNA molecules. So this is normal DNA molecule and this is hybrid DNA and this one is also a hybrid DNA and this is normal DNA molecule. This normal DNA as well as this hybrid DNA both are formed in the equal proportions. Is it clear? This experiment a little bit messy and confused. So all of you listen carefully. So this experiment is conducted in the E. coli cell, and these E. coli cells are cultured in the medium with N15 isotope. After the completion of the one generation, it can give two daughter cells with normal DNA and N heavy DNA, and they again undergo another generation. It can give four daughter DNA molecules. Out of four daughter DNA, one is normal and the another one is normal, and these two are hybrid DNA molecules. This experiment is clearly indicates that DNA replication takes place through semi-conservative method, and these strands are separated with the centrifugation with cesium chloride solution. Okay, it is about experimental proof by Michelson and Stahl in E. coli bacteria. Okay, the total DNA replication process as well as experimental proof is completed. Then let us discuss about packing of DNA helix. Packing of DNA helix means the length of the DNA is sometimes more than the cell size. So that's why the DNA molecule is closely packed inside the nucleus. Okay, so in the case of prokaryotes, any idea about prokaryotes? Prokaryotes means the nucleus is not bounded by nuclear membrane. Prokaryotic cell means nucleus is not bounded by nuclear membrane. The best example for prokaryotic cell is bacterial cell. So, in this prokaryote cells, at the place of nucleus, only one DNA molecule is present. This DNA molecule is not bounded by nuclear membrane, and this cell is called as prokaryote. Pro means primitive, karyote means nucleus. Primitive type of nucleus is present. Okay, so in the case of prokaryotes, the DNA of prokaryotes is almost circular and lacks the chromatin organization. This is referred to as genophore. Genophore means in the case of bacteria, in the place of nucleus, only the genetic material DNA is present with it is in the case of eukaryotes. Eukaryotes means the nucleus surrounded by well-developed nuclear membrane. So it is the plant cell or animal cell. First we take the plant cell. So in the middle portion of the cell is occupied by nucleus, and this is the nuclear membrane. Inside the nucleus, DNA is present. So eukaryotes, U means advanced, karyote means nucleus. Advanced type of nucleus means well developed nucleus is present in the case of eukaryotes. Well developed nucleus means nuclear genetic material DNA is covered with well developed nuclear membrane. Nothing but nucleus is bounded by nuclear membrane. Okay, so in the case of this eukaryotes, this this organization means packing of DNA is more complex. Okay, there is a set of positively charged basic proteins called as histone proteins are present in these eukaryotes. Listen carefully. In the case of prokaryotes, there is no histone proteins and there is no chromatin organization because its nucleus is poorly developed. Only genetic material DNA is present, so that's why there is no chromatin organization. But in the case of these eukaryotes, well developed nucleus is present. Nucleus is bounded by nuclear membrane, so that's why this packing of DNA is much complex, and this DNA molecule is negatively charged, and this DNA molecule is paired with some basic proteins. They are positively charged basic proteins called as histone proteins. Histone proteins are basic in nature. DNA is acidic in nature, and this DNA molecule is negatively charged. DNA is acidic in nature and it is negatively charged. And these proteins are basic in nature, they are positively charged. Okay, and these histones are rich in basic amino acids residues. They are lysine and arginine. These two amino acids are present in the histone proteins. Is it clear? In the case of eukaryotes, histone proteins are present, and these histone proteins are basic in nature. They consist basic amino acid residues like lysine and arginine. This both the amino acid residues carry positive charge in their side chains. Is it clear? These amino acid residues they can carry the positive charge in their side chains. And these histones are organized. These histones are organized to form a unit of 8 molecules called as histone octamer. So this is histone octamer. Histone octamer means this DNA. This is DNA molecule, and these are the histone proteins. So, and this DNA molecule is wrapped around the these histone proteins are wrapped around the DNA molecule. They can form the octamer, histone octamer core. Listen carefully. The negatively charged DNA is wrapped around the positively charged histone octamer to form a structure called as nucleosome. This structure is called as nucleosome. Nucleosome means negatively charged DNA. This negatively charged DNA is wrapped around the histone octamer to form a nucleosome. So DNA molecule is negatively charged, which is acidic in nature. These histone proteins are positively charged, they are basic in nature. So this negatively charged DNA is wrapped around the histone proteins and they can form the nucleosome. It is histone octamer. Histone octamer means there are 4 types of histone proteins are present in eukaryotes. They are H1, H2A, H2B, H3, and H4. These are the 4 types of histone proteins which are present in the eukaryotes, and they can form the histone octamer. And this DNA is wrapped around the histone octamer to form a nucleosome. And a typical nucleosome contains 200 base pairs of DNA helix. And this nucleosome constitute the repeating unit of the structure in the nucleus called chromatin. Listen carefully. This nucleosome constitute the repeating units of the structure. The nucleus is called as chromatid. So this nucleosome in the chromatid are look like a beads on a string-like structure when we observe under the electron microscope. So when we observe the chromatin, in this chromatin, some bead-like structures are there. This is the chromatin, nothing but thread-like structures, nothing but chromosomes. So in this chromatin, some beads like structures are present. The nucleosome in chromatin are seen as beads on string like structure. These beads on string like structure are viewed under electron microscope. These beads on string like structure are observed under electron microscope. Okay, so this chromatin at the time of cell division, it is nothing but mitosis or meiosis. Mitosis is takes place in the vegetative or somatic cells. Meiosis is takes place in the diploid reproductive cells. It results in the formation of gametes, male and female gametes. So, at the time of cell division, this chromatid is condensed to form short rod-shaped structures called as chromosomes. This chromatin, this chromatin at the time of cell division is condensed to form these short rod-shaped structures called as chromosomes. So it is about packing of the DNA. So in the case of eukaryotes, this chromatin is two types, euchromatin as well as heterochromatin. Euchromatin means it takes less amount of stain and it appears light in color. It is called as euchromatin. Euchromatin means the chromatin that is more densely packed and takes more stain. Okay, chromatin is again two types, euchromatin and heterochromatin. Heterochromatin means it is loosely packed and takes dark stain. It is called as heterochromatin. Euchromatin means the chromatin packs loosely and takes little stain. It is called as euchromatin. Is it clear? Chromatin is two types, euchromatin and heterochromatin. Euchromatin means it takes less amount of stain and it can loosely packed. It is called as euchromatin. And heterochromatin means it takes more stain, it is appear in dark color and it is very tightly or densely packed. That region is called as heterochromatin region. And functionally, euchromatin is active. Euchromatin is active and which is having more functional genes and it is transcriptionally active chromatin. This euchromatin is transcriptionally active chromatin. It having more functional genes, where heterochromatin is inactive and it consists non-functional genes. Okay, it is about total packing of DNA. Okay. Next, one genetic material DNA explanation is totally complete, and the another type of nucleic acid is RNA. Second type of nucleic acid is RNA. RNA full form is ribonucleic acid. So this is RNA full abbreviation or full form, that is ribonucleic acid. It is a polymeric molecule essential in various biological activities or roles and it is involved in coding, decoding, regulation and expression of genes. Okay, RNA, it is also another type of nucleic acid Little bit of RNA is present in the nucleus and more amount of RNA is present in the cytoplasm. Is it clear? Very little bit, 10 to 15% of RNA is present in the nucleus, and the remaining portion of RNA is present in the cytoplasm of the cell. And this RNA in some viruses it is functions as genetic material, and in most of the organisms it is acts as a catalyst, and it also functions as messenger as well as adapter. Like DNA, RNA is assembled as a chain of nucleotides. But unlike DNA, this RNA is found in nature as a single strand folded onto itself rather than a paired double strand. DNA is present in this double helical structure, but in the case of RNA is present only in a single strand. But itself it fold to appear a pseudo helic like structure. Basically RNA it consists only one single strand, but in DNA it consists two strands. In the case of RNA it consists only one single strand. In many viruses RNA is functions as genetic material. In some viruses like retrovirus, in that retrovirus RNA is functions as genetic material. So next, the nitrogen bases in the RNA are adenine, guanine, cytosine and uracil. This uracil which replaces the thiamine in DNA. In previous class we already discussed about the nitrogen bases. Two types of nitrogen bases are there, purines and pyrimidines. Adenine, guanine are purines, thiamine, cytosine are pyrimidines. So in the case of this RNA, adenine, guanine both are purines which are present in the RNA. Next, coming to the pyrimidines, cytosine and uracil are present. In the place of thiamine, uracil is present in the RNA. This uracil, which replaces the thiamine in DNA, means in the place of thiamine, uracil is present in the RNA molecule. RNA is typically a single-stranded biopolymer. I already explained now. This RNA is a single-stranded biopolymer. Okay. This is the structure of RNA. This RNA consists of four nitrogen bases. They are cytosine. This is cytosine and this is guanine, and this one is adenine and last one is uracil. Uracil is present in the case of RNA instead of thiamine. So this RNA molecule is single-stranded molecule. These are the nucleobases, nothing but nitrogen bases. These base pairs are present in the helix of sugar phosphate. RNA molecule is the single-stranded biopolymer. It does not consist double strand. It consists only one single strand. Next, coming to the types of RNA. There are three major types of RNAs are there. They are mRNA, rRNA and tRNA. There are three major types of RNAs are there. They are mRNA, nothing but messenger RNA. mRNA means messenger RNA. Second one is rRNA. rRNA means ribosomal RNA. And the last, third one is tRNA. tRNA means transfer RNA. These three are the three major types of RNAs. Let us discuss about mRNA, nothing but messenger RNA. This mRNA transcribes the genetic code from the DNA into a form that can be read and used to make the protein. All of you listen carefully. This mRNA is synthesized from the DNA molecule through transcription process. Transcription means synthesis of mRNA from DNA. That process is called as transcription. The transcription process is discussed on another session in detail, but the definition is synthesis of mRNA from DNA is called as transcription. And this mRNA transcribes the genetic code from the DNA into a form that can be read and used to make a protein. This mRNA, it is plays an important role in protein synthesis process. This mRNA carries the genetic information from the nucleus to the cytoplasm of the cell, which means the synthesis of mRNA takes place inside the nucleus because DNA molecule is present inside the nucleus. From that DNA, this mRNA is synthesized. That synthesized mRNA, it carries the genetic information from the nucleus to the cytoplasm because in the cytoplasm, protein synthesis process is takes place. Protein synthesis processes takes place in the cytoplasm and the ribosomes. Ribosomes are sites for protein synthesis. So this mRNA, which carries the genetic information from nucleus to the cytoplasm of the cell. This is about mRNA. Next, the second type of RNA is rRNA. rRNA means ribosomal RNA. This rRNA is located in the cytoplasm of a cell where ribosomes are found. Means this rRNA is present on the ribosomes. These ribosomes are one of the type of cell organelles. They are present in the cytoplasm. This rRNA directs the translation of mRNA into protein. Is it clear? This rRNA, it gives directions to the mRNA in the translation process to synthesize proteins. It is about rRNA. Next, last type of RNA is tRNA. This is tRNA. This tRNA is located in the cytoplasm of the cell. This tRNA is located in the cellular cytoplasm and it is involved in the protein synthesis. This tRNA, it functions as adapter and it consists one amino acid attachment site and another anticodon region. It functions as adapter molecule and which is involved in the protein synthesis. tRNA means transfer RNA brings or transfers the amino acids to ribosomes that corresponds to each three nucleotides of the codon of rRNA. The amino acids can be joined together and processed to make a polypeptide chain with proteins. All of you listen clear or carefully. This tRNA is located in the cytoplasm, which is plays an important role in the protein synthesis. This tRNA, it transfers the amino acids to the ribosomes. Ribosomes are sites for protein synthesis, and this tRNA transfers the amino acids to the ribosomes correspond to three nucleotides of the codon of rRNA. And these amino acids then can be joined together with the help of polypeptide bonds to form a polypeptide chain with proteins. It is about tRNA. Okay. Next, coming to the assignment 1. This is the first assignment. First question is, What are the components of a nucleotide? Okay, nucleotide consists three major components. We already discussed in the previous class. They are nitrogen bases, pentose sugar, and phosphate radical or phosphate group. They are constitute together, they can form a nucleotide. It is the first question. And the second question is, what is the function of DNA polymerase? In the today's class, we have discussed about the DNA polymerase role in the DNA replication process. Next, the third question is, what is the differences between exons and introns? Okay, next, coming to the fourth question, that is, write any two differences between DNA and RNA. Write about the two differences between DNA and RNA. All the students complete this assignment and send to your concerned JLs. Okay, thank you. Today's session is completed. వాడిని ఎందుకు తీసుకొచ్చినవరా? అన్నం తిన్నాక రాకపోతుండేనా? తాత ఏం చేస్తాడు?
Completed, madam. Madam,